plasmid. So plasmid is a small extra chromosomal DNA which can replicate independently. It was termed by the biologist Joshua Lederberg in 1952. So you can see here in this picture, this is a bacterial cell in which there is a main bacterial DNA along with which extrasomal plasmid is there. They are mostly found in bacteria. Some of the eukaryotes like yeast and plants also contain plasmids. Plasmids have a wide range of lengths from roughly 1000 DNA base pair to 100,000 of DNA base pairs. Properties, they are specific to only few particular bacteria, replicate independently, and can code for their own transfer. Do not cause damage to cell and generally are beneficial, do not have extracellular forms and exist inside cells simply as free and typically circular DNA. Size range from 1 kbp to several mbp. Number of plasmids in an individual cell may vary, ranging from 1 to several hundred, denoted by a copy number. Genes carried by plasmids encodes traits for antibiotic resistance or resistance to heavy metal. Some produce virulence factors that help in defense or nutrient utilization. Plasmids can also have bacteria with ability to fix nitrogen. What are the elements of plasmid? So a plasmid has origin of replication. Here we can see it is the DNA sequence which initiates the replication, antibiotic resistant gene site, which provide them antibiotic resistance for their survival in the host. It also had multiple cloning site. It is a short segment containing several restrictions enzyme site, enabling easy insertion of foreign DNA. Insert. It is a foreign DNA cloned in multiple cloning site. Promoters are it drives the transcription of the foreign insert and selectable markers. It is used to select the cells that have successfully taken up the plasmid for the purpose of expressing the insert. Primary binding site. It is the site for binding the short single-stranded DNA sequence used as the initiation point of the PCR amplification or sequencing of the plasmid. Plasmid. They replicate autonomously because they have their own replication origin. Most plasmid in gram-negative bacteria replicate in a manner similar to the replication of bacterial chromosome, involving initiation at replication around the DNA circle, giving a theta intermediate. Most plasmid of gram-positive bacteria replicate by a rolling circle mechanism. Mode of transfer of plasmid, or the genetic information encoded in a plasmid of bacteria transferred across a broad range of microorganisms via transformation, transduction, and conjugation. Here we can see the three mode of transfer of plasmid by transformation, transduction, and conjugation. In transformation, a competent cells is required, which is ready to accept extracellular plasmid and for further replication in the host cell. In transduction, the plasmid-mediated gene transfer through bacteriophage it can be specialized or generalized transduction. In conjugation, cell-to-cell contact is required of donor and recipient, which requires for DNA metabolism of donor cells. Classification based on their function and plasmids are classified as follows: F plasmid or fertility plasmid. It contains TRA genes or transfer genes. They are capable of conjugation and result in expression of sex pili. R plasmid or resistant plasmid. It contains gene that provides resistance against antibiotic or antibacterial agents. Col plasmid. It contains genes that code for the bacteriocins protein that can kill other bacteria. Degradative plasmid. It helps the host bacterium to digest compounds that are not commonly found in nature, such as camphor, xylene, toluene, and salicylic acid. These plasmid contain genes for special enzymes that break down specific compounds. Degradative plasmid are conjugative. Virulence plasmid. When a virulence plasmid is inside a bacterium, it turns that bacterium into a pathogen which act as an disease-causing agent. So here we can see the example of PBr 322 plasmid. It is a artificially made plasmid. It has relatively small size of 4363 363 base pairs. The nomenclature is the P stands for plasmid and BR stands for the researchers who developed it, Bolivar and Rodriguez. 322 is the order of its invention, just like any other plasmid, to differentiate like PBR 325 and PBR 327. The plasmid, this plasmid was invented or created in 1977 in the laboratory of Herbert Boyer at University of California in San Francisco. This plasmid is 4361 base pair and has two antibiotic resistant genes for ampicillin and tetracycline. It has genes of ampicillin resistant and tetracycline resistance. The gene BLA is encoding for the ampicillin resistance protein, and the gene TET-A, or that is tetracycline A, is encoding for tetracycline resistant gene. And this the we can see here are the restriction sites. Use of PBR 322. It is widely used as cloning vector. In addition to this, it has been widely used as model system for the study of prokaryote transcription and translation. Thank you. Yes, sir. Mostly in prokaryotes, there are circular plasmids and linear plasmids. It can be, I think, or for like in eukaryotes, some. Yes, sir. Yes.
Yes, sir. Sir, थोड़ा screen share. Share. कैसे करते हैं? हाँ, किया, सब बंद किया. Okay. हाँ, अभी दिख रहा है स्क्रीन विजिबल है? हाँ, है ना? आया पीपीटी? हेलो?
No, sir.
नहीं दिख रहा है? सर, हाँ. सर, हेलो? हेलो? हाँ सर, आ रहा है? सर नहीं हो रहा है। सर मैं आपको सेंड करूँ, आप ये करोगे प्लीज? हो गया सर, मेरा हो गया बिल्कुल। कर दिया आपको सेंड। हाँ देखो, सेंड किया। हेलो सर। परफेक्ट। ओके, गुड इवनिंग एवरीवन। आ रहा है आवाज सर? हेलो सर, आपका माइक ऑफ है। अपन, हाँ, yeah. Good evening, everyone. My name is Karthik Chavan, roll number 227307, and my topic is restriction mapping. Next page. Yeah. So before moving into restriction mapping, I would like to introduce some of the basics that are restriction enzymes. So what are restriction enzymes? Restriction enzymes are one of the most important tools in the DNA, that is the recombinant DNA technology. Whereas so these are the DNA cutting enzyme present in bacteria that is prokaryotes that recognizes the specific sites in the DNA. So there are various DNA cutting enzymes which have the ability to recognize a specific specific sites in the DNA. So that the specific sites are called as restriction sites and make our DNA double strand break at or near recognition sites. So when the cut is made in the restriction sites, the newly produced DNA end will have either a blunt end or a sticky end. That is when the restriction enzyme acts on a DNA. So the cut is formed. So in that cut, there are two cuts will be formed. That is the blunt ended and the sticky ended. So the diagram you can see over, the first one is the blunt ended. That is it's blunt. Uh, and then the other one is the staggered end, also called as the sticky ends. So yeah, second one. So you can see over there like PTAA can stick to the upper stand, that is AATPC. So it's staggered cuts. Next. So restriction enzyme slash endonuclease, also known as a molecular scissor. So there are normally three types of restriction enzymes that are type 1 restriction enzyme, type 2 restriction enzyme, and type 3. And the fourth one is the type 4. So in the type 1 restriction enzyme, so moving into the definition of type 1 enzymes, they are complex multi-subunit combination restriction and modification enzymes that cut DNA at random far from their recognition sequence. So there are specific sites where the what we say where the restriction enzyme act. So or we can say that there are some recognition sequence where the cuts have to be made. What restricts type 1 enzyme do is far away from the recognition sequences, they make the cut. The second one is type 2 enzymes. So cut DNA at the defined position close to or within the recognition sequence. So the most accurate one is the type 2 restriction enzyme. So what they do is they cut the defined position, means where they have to cut at a specific spot, they cut on it or besides the recognition sequence. The third one is the type 3 restriction enzymes. So they cleave outside of the recognition sequence. That is, for example, if there is a restriction enzyme, they do not cut on or within. They cut the outer side of the recognition sequence and require two subsequences in opposite orientation within the same DNA molecule to accomplish cleavage. They rarely give complete dialysis. The next one is type 4 enzyme. So they recognize modified. There is a modified DNA sequences, typically methylated DNA and cleave in at that region. For example, if there is a DNA sequence and the methylated group is there, so what the type 4 enzyme do is they cleave at the methylated DNA and cleave at that region. Next slide, please. Next slide. Yeah. So as I explained the basics, I would like to move into the restriction mapping. So what is restriction mapping? So it is a method used to map an unknown segment of a DNA by breaking it into the pieces and then identifying the location of the breakpoints. So, in a simple term, what we can say is, say, it is a method to map or to locate unknown segment of a DNA. So, in order to locate the unknown segment of a DNA, what we can do is we can consider that segment, break it into pieces, and we can identify the location of the breakpoints. So that is in simple term is known as restriction mapping. So this method, the restriction mapping, so relies upon the use of protein called restriction enzyme. So for this method, for restriction mapping, of course, they require the proteins called as restriction enzymes. So what are the function of the restriction enzymes? We'll look earlier is that they cut, they digest. DNA molecules are short, of course. So specific sequences called restriction sites. So at specific recursion sites, what restriction enzyme do is they cut they what we say they digest the strands. After DNA segment has been digested using a restriction enzyme, the resulting fragments can be examined using a laboratory method called gel electrophoresis. Once the work of the restriction enzyme is done, that is by digesting or cutting the DNA segment, the next task is that the DNA fragments that are formed we can do the technique called electrophoresis in order to identify, which is used to separate pieces of DNA according to their size. So the pieces will be separated by electrogel of what we say, electrophoresis, gel electrophoresis. We can separate according to their size. So next point, one common method for constructing a restriction map involves digesting the unknown DNA sample in three ways. So in order to map the DNA segments, so there are three ways. Here, two portions of the DNA sample are individually digested with different restriction enzymes, and the third portion of the DNA sample is double digested with both restriction enzymes at the same time. The next point, next, each digesting sample is separated using gel electrophoresis. The sample we obtain, we have to we have to separate it using gel electrophoresis, and the sizes of the DNA fragments are recorded. So as soon as the fragments are separated, we can record these sizes that how much a kilodalton the size we have obtained by using gel electrophoresis. So the total length of the fragment in each restriction will be equal. However, because the length of each individual DNA fragment depends upon the position of the restriction site, each restriction site can be mapped according to the length of the fragment. And then what they're saying is that the position of the restriction site, each restriction site can be mapped. Uh, each restriction site can be mapped according to the length of the fragment. So, depending upon the length of the fragment, we can map the restriction site also. Next slide, please. The information from the double digestion is particularly useful for correctly mapping the sites. The final drawing of the DNA segment that shows the position of the restriction site is called a restriction map. Next point is it is possible to determine the position of the restriction site on a DNA fragment by digesting it with various restriction enzymes singly and in combination and analyzing the fragment sizes obtained. Next, restriction mapping used to be a procedure for characterization of a cloned fragment of a DNA, but this is now more easily done by DNA sequencing. So the what we say, let's say restriction mapping and all, so it can be easily done by the newest evolving method called DNA sequencing. Nevertheless, analysis of restriction enzyme or the fragment sizes is still useful. For example, in comparing the chromosomal organization of different strains. In order, if there are different strains of a species, so in order to differentiate them, we can use the mapping, that is restriction mapping, in order so that we can compare their fragment sizes with each other. Next, please. So the what is the process of restriction mapping? Is the sample first we have to take the sample DNA. We have to digest it using the restriction enzyme. We can use type 1, type 2, type 3, or type 4. So if you want accuracy, we can go for type 2 restriction enzyme. After that is the restriction digestion. So the DNA sample we obtain is single. So we have to digest it so that the ones which are double stranded we can digest it and turn into single fragment. Then after the fragments and all is formed, full single or double, we can use the agar gel electrophoresis. So what is the main function of the agar gel electrophoresis is that once these broken fragments are obtained, what we can do is we can separate it according to the size. The next one is DNA pattern and band visualization. By staining, we can perform the band visualization as we have learned earlier in the blotting techniques and all, electrogel, electrophoresis and all, how the staining is done. After the staining, we can go for band analysis. That is which band has which position, what is the weight, what is the size of the DNA fragment. And that is how the restriction map generation is done. Next, please. Yeah. So, yeah, looking into a diagram. So there is a 12 kilo base DNA segment to be mapped. Okay, so consider this is an example. Okay, so there are three techniques. As during explanation, you might have noticed that I gave the what we say, I gave three example. Okay, yeah, three example. So there are single digest second, and the double digest. So in the single digest, that only one restriction enzyme is used, and in the double digest, two enzymes are used. So we can look at the binds. At the first one is that 3 kilobase pairs and 9 kilobase pairs. Second one is the 5 kilobase pair, 7 kilobase pair, and the third one is the double digest. That two enzymes are used. You can see uh, two base pair, 3 kilobase pair, and 7 kilobase pair. Three strands are formed. So for taking about the single digestion, E coli one is used. The second BAMH1 is used, and the both enzymes are used in the double digest. That is, the both enzymes are used simultaneously. So what we can see is yeah, please. application application of restriction enzyme. So we have learned about the basics. We have learned about how the perform is done, how the restriction in the mapping is done. Now let's move on to the application of restriction mapping. So what is the first is that identification of restriction type. So in order to identify the restriction type, DNA mapping can be used. The second one is insertion and deletion. This is the study of a gene. So in order to insert a modified gene or the selective gene, we can use the restriction mapping. The third one is the insert analysis during cloning. So we can analyze what we are inserting during the cloning test. The fourth one is verification of the size of the insert during cloning. So when we are performing the cloning, we can verify the what size of the DNA fragment we are introducing in a selective DNA fragment. And the last and final is the mutation studies, of course. Next, please. Thank you. I would like to end my presentation. Any doubts or questions, please? सर नहीं समझा फिर से एक बार बोलो मैं ओके ओके दो रेस्ट्रिक्शन दो साइट है दो एंजाइम है और एक ही सिंगल इसको कट मारे तो उसको क्या बोला जाता है नहीं मालूम सर सॉरी न्यूसिजोमस हेलो so न्यूसिजोमस so the term news correct meta न्यूसिजोमस ना नहीं मालूम सर सॉरी हाँ 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 Sir, methylation, na, so in order to, for example, methylation is kind of a tagging system for a particular bacteria. So we can say that methylation में sir मिथाइल ग्रुप ऐड करते हैं एक पर्टिकुलर इसके लिए सर हाँ सो मिथाइल डीएनए को मिथाइल ग्रुप ऐड करते हैं उससे क्या होता है सर जो खुद के काइंड ऑफ टैग सिस्टम यूज करते हैं फॉर एग्जांपल दे टैग देमसेल्व्स सो इन ऑर्डर if they are introduced, so they will be able to recognize कि like हाँ ये अपना ही बंदा है करके. So they won't act on him. Instead of they would act on the foreign affairs. तो सिंगल डाइजेस्ट में ना सर एक ही रेस्ट्रिक्शन एंजाइम यूज करते हैं, वैसे डबल डाइजेस्ट में ना दो रेस्ट्रिक्शन एंजाइम यूज करते हैं एक ही जगह को क्लीव करने के लिए. हेलो, थैंक यू सो मच सर. बाय.
Yes, sir. Yes, sir. Sir, it is visible. Hello, sir. He- Okay, sir. Hello, everyone. Good evening. My name is Sakshi Bansi Amle, roll number 227302, and my topic is restriction enzyme. What happened? Sir, where it is? Sir. Sir. So here is the definition of restriction enzyme. A restriction enzyme is a protein isolated from bacteria that cleaves DNA sequence at sequence specific sites with producing DNA fragments with a known sequence at each end. The use of restriction enzyme is critical to certain laboratory methods and including recombinant DNA technology and genetic engineering. In simply, we can say it is the molecular scissor for the cut to cut double strand DNA molecule at a specific points, and it is found naturally in a wide variety of prokaryotes, and it is important tool for a manipulating DNA. So these enzymes are found in bacteria and archaea are provide a defense mechanism against invading viruses. Inside a prokaryote, these restriction enzymes selectively cut a foreign DNA in a process called restriction digestion. Meanwhile, host DNA is protected by a modification enzyme that is a methyl transferase that modifies the prokaryote DNA and blocks cleavage. Together, these two processes form the restriction modification system. More than 3,600 restriction endonucleases are known, which represent over 250 different specificities. Over 3,000 of these have been studied in detail, and more than 800 of these are available commercially. So, these enzymes are routinely used for DNA modification in laboratories, and they are vital tool in molecular cloning. So, what are restriction enzymes? So, these enzymes is a protein produced by bacteria that cleaves the DNA at specific sites, or these sites is also known as the restriction sites. The restriction enzyme protect the live bacteria from bacteriophage, and they recognize and cleave at the restriction sites of the bacteriophage and destroy its DNA. So the restriction enzymes recognize short and specific nucleotide sequence in the DNA known as the recognition sequence. And when the restriction enzyme recognizes a DNA sequence, it hydrolyzes the bond between adjacent nucleotide and cuts through the DNA molecule. So means what? Each restriction enzyme recognizes a short specific sequence of nucleotide bases the four basic chemical subunits, that is the linear double stranded DNA molecule are made from a nucleotide bases, that is adenine, cytosine, thymine, and guanine. And to cut their DNA substrates at their recognition sites if the recognition and cleavage sites are separate from another. So to cut DNA, all restriction enzymes makes two incisions, means one through each sugar phosphate backbone and each strands of the DNA, that is double helix. And in this means prevents it prevents live bacteria from the restriction endonuclease. Methyl, it is an important role is a methyl transferase, means what is the role? It is methyl groups. Methyl groups are added same sites and the protects the host genome from degradation by the restriction enzyme nuclease. There are four types of restriction enzymes. Type 1 restriction enzyme, type 2, type 3, and type 4. So type 1 is a restriction enzyme, are also called as restriction endonuclease. They are made up of two long strands of DNA joined together. These restriction enzymes recognize certain sequences of DNA and cleave them at a site. Type 2, it is made up of four strands of DNA, two on each side of the DNA. They usually recognizes two bases on one strand of DNA and cleaves the DNA. Type 3 means restriction enzymes are DNA cutting enzymes that recognizes pattern of DNA that are not necessarily based on DNA sequence. And type 4, it is methylation dependent restriction enzymes, MDRs are RES that preferentially target modified DNA containing glycosylated bases or methylated on adenine or a cytosine residues but lack a cognate M test altogether. Here it is a picture of sticky ends, and a restriction enzyme is a DNA cutting enzymes that recognizes specific sites in DNA. Many restriction enzymes that stagger cuts, also called as sticky end, or near their recognition sites, producing ends with a single stranded overhang. Many restriction enzymes make staggered cuts, producing ends with single-stranded DNA overhangs. However, some produce blunt ends. DNA ligase. DNA ligase is a DNA joining enzyme. If two pieces of DNA have matching ends, ligase can link them to form a single unbroken molecule of DNA. In DNA cloning, restriction enzymes and DNA ligase are used to insert genes and other piece of DNA into plasmid. And here it is a picture of blunt end. Blunt end means what? It cut both strands of equal length, and they end at the same base position, having no unpaired bases in either strand. And sticky ends means it cuts in a staggered, and one strand is longer than the other, typically by at least a few nucleotides, but such that the longer strand has bases which are left unpaired. So here it is the examples of how restriction enzyme cleave DNA. That is, as SMA1 results in blunt ends, BAM-H1 results in 5 overhanging sticky ends, and C, it is a PST1 results in 3 prime overhanging sticky ends. So cut with SMA1, here it is a blunt end, so it is cut in the middle and cut in equal length, and both sides pairs are equal. And second one is cut with BAM H1, uh, 5 overhang, it is a 5 prime overhanging sticking end, and it is staggered cuts. In 5 prime, GATCC is cut, and in 3 prime only end of G is cut, and in 5 prime C, the C, double GATC is remaining. No, G is cutting, and GAT double CC is remain. And in 3 prime to 5 prime, double CTA cut, and G is remaining here. And in C, it is a 3 prime overhanging. So in that मतलब फर्स्ट वाला जो कट होता है एंड बाकी रिमेन इट और इन 3 prime to 5 prime G is remaining here and ACGTC are cut there, and in 5 prime to 3 prime G at one end G is remaining and CTGCA is cut in here, and in 3 prime to 5 prime C is A G in first G is cut there and ACGTC are remaining here. So here are some examples of recognition sites of representative restriction endonuclease. So enzyme and their source and recognition site are there. So in BAM H1, the name of or is Bacillus amylocufacins H, and recognition site is double G8TCC. E core 1 Escherichia coli RY13, recognition site is GAATTC. Hay 3 Haemophilus aegyptus, recognition site is double GCC, and Hin 3 it is Haemophilus influenzae RD, recognition site is double AGC HPF1, this is Haemophilus influenza, recognition site is GTTAC. In HPA22 Haemophilus parainfluenza it is also, and recognition site is double C double G. MBO1 it is Moraxella bovis, recognition site is GATC. Not one, it is a Nocardia otidivis cavarium, and recognition site is GC double G double CGC. SF51, this is Streptomyces fambreatus, recognition site is double G double C 5 times N and double G double C. And tag one Thermus aquaticus, it is recognition site is TCGA. So there are some enzymes and their functions. So restriction endonuclease type 2, the function is cleaves DNA at specific base sequence. Second one is DNA ligase. The function is joins means it joins the two DNA molecules. DNA polymerase 1, it is addition of nucleotide at 3 prime ends of duplex DNA molecule for filling gaps. Reverse transcriptase function is makes a DNA copy of an RNA molecule. Then fifth one is polynucleotide kinase. It is function is add a phosphate to 5 prime OH end of a polynucleotide. Sixth, terminal transferase, and their function is adds homopolymer tails to the 3 prime OH ends of a linear duplex. Exonuclease 3, if their function remove nucleotide from the 3 prime ends of a DNA molecule. Bacteriophage 1 exonuclease enzyme, and their function is remove nucleotide from the 5 prime ends of a duplex to expose single stranded 3 prime ends. And last one enzyme is alkaline phosphate, and their function remove terminal phosphate from either the 5 prime or 3 prime end or both. So discovery of restriction enzyme. So Werner Arber started this field of research in Geneva during the 1960s. So he discovered restriction enzyme. Arber was studying an earlier known phenomenon, the host control restriction of bacteriophages, and found that this process involves change in the DNA of the virus. So there are some applications of restriction enzyme. So first one is means it is used in gene cloning and protein expression experiments. Second one is restriction enzyme that are used in biotechnology to cut DNA into smaller strands in order to study fragment length difference among individuals. That is RFLP, known as restriction fragment length polymorphism. And third one is each of these methods depends on the use of agarose gel electrophoresis for a separation of the DNA fragments. Thank you. Uh, So it means different restriction enzyme that recognize the same sequence. It is known as neosesomers. Uh, Isothesomer means that a different enzyme cleaves the same location. Difference between means in neosesomers that means different enzymes जो same sequence that means recognize the same sequence और in isosomers में जो different enzymes जो cuts in same location पे वो cut करते हैं और in sir repeat the question. सर फॉर्मूला बताना है वैल्यू sequence. हाँ सर मतलब GC50 है अब सर सबका रेशियो 1 आता है ना सर कुछ सर रेशियो फॉर्मूला ना सर रुको याद करने दो सर सर्च नहीं कर रही हूँ सोच रही हूँ एडिनिन सर मतलब
Yes, sir. Yes. Is it visible? Hello. Good evening, everyone. My name is Aisha Mullah, and my roll number is 227304. And today, my topic of presentation is bacteriophage and properties of one of the bacteriophage. First, I will start with a small introduction with viruses. So, virus is a submicroscopic entity, which means it is too small to be seen with the help of a microscope. Viruses are infectious agent which affects all life forms. Two scientists contributed to the discovery of virus. First was Bejernik. He was the scientist who discovered a pathogen that was smaller than a bacterium from tobacco mosaic plant and called it a virus. And therefore, he is also known as father of virology. And Ivanovsky was a Russian botanist and co-discoverer of viruses. And the study of virus is known as virology. Now, introduction to bacteriophage. Bacteriophages are commonly called as a phage, it is a virus that infects and replicates only within the body of a bacteria. The genetic material consists of either DNA or RNA, which is encapsulated in the head region made from a protein coat. And bacteriophage was discovered independently by Frederick Twort in Great Britain and Felix de Hull in France. Uh, de Hull coined the term bacteriophage, which means bacteria eater. In the the picture depicts how bacteriophage looks like. Moving on to the structure of bacteriophage, as you can see in the diagram, the bacteriophage is basically made up of three important parts. First is a polyhedral head, a short collar, and third, tail fibers. The head is basically made up of protein coat known as capsid. It helps to carry the genome of the virus. The head may be enveloped or non-enveloped, which means enveloped viruses have a phospholipid membrane outside the capsid, whereas non-enveloped viruses do not have a phospholipid membrane. The collar and whiskers help to attach six long tail fibers to the phage particle during the virus assembly, and the tail fibers help in attachment of the virus particle to the bacterial cell wall. Bacteriophages to make number of copies, the bacteriophage follow the replication in either two ways, either through lysogenic cycle or lytic cycle. First, I will start with the lytic cycle. In lytic cycle, the phage causes complete lysis of the bacterial cell. Examples like T2, T4, etc. They are also known as virulent phages. The cycle has following step. First step is the virus recognizes the host cell and anchors to the bacterial cell wall with the help of tail fibers. The second step is penetration, where phage DNA gets injected into the bacterial cell. Third step is replication. The bacterial DNA is disrupted and the viral genome takes over the cell's protein synthesis and DNA replication machinery, forcing it to express phage genes and replicate the phage DNA. Fourth step is assembly. Here, the phage components are assembled into phage particles. The fifth step is lysis and release. Bacterial cells are lysed and new viral particles are liberated to infect other cells. The lytic cycle is also known as reproductive cycle of the bacteriophage. Next is lysogenic cycle. In lysogenic cycle, the viral DNA gets integrated into the host genome and replicate along within the bacterial genome. Firstly, the virus attaches itself to the host bacterial cell and gains entry by injecting its DNA into the bacterial cytoplasm, as you can see in the diagram. In the third step, is integration, where the viral DNA gets integrated with the bacterial DNA. The viral genetic material is read and replicated by the same bacterial proteins that replicate bacterial genetic material. Now, the integrated genetic material is called a prophage. After a prophage is formed, it switches to lytic cycle, which means the bacterial mechanisms are used to produce lots of DNA and lots of capsids and protein covers. So the capsid gets released into the environment to infect new bacteria and the lysogenic cycle may start again. My next subtopic is properties of one of the phages. So I have chosen lambda phage. Lambda phage is also known as Enterobacteriaphage lambda. It is a bacterial virus that infects the bacterial species of Escherichia coli. The virus is template, which means the infection does not immediately result in cell death. The viral genetic material remains dormant. The lambda phage infects other E. coli bacteria by the process of specialized transduction. It is a process by which only restricted set of genes is transferred to the cell. And this phage was discovered by the scientist Esther Lederberg in 1950. The genetic material of the lambda-phage belongs to the family of Cephuviridae, and the genetic material can be found in the head part of the virus. A double-stranded DNA structure serves as the genetic material, and the virus is also made up of head, tail, and tail fibers. The It is non-enveloped and has a non-contractile long tail. This virus undergoes both lytic and lysogenic cycle, and it is commonly used as a vector in recombinant DNA technology. Now, applications of bacteriophage. First is in treatment of bacterial infection. With the increasing cases of bacterial resistance against numerous antibiotics, the potential use of bacteriophage treatment has been explored. As the bacteriophage infects only bacteria and is harmful to the administration, and it is harmless to humans. The administration of such bacteriophage into humans helps in destruction of such infectious bacteria. Second is environmental sanitation of hospitals. Many hospital acquired infections can be transmitted by pathogens contaminating hospital surfaces, which is not efficiently controlled by conventional sanitation. Bacteriophage have been suggested as decontaminating agents based on their selective ability to kill specific bacteria. These things can be also be employed in industries also. Third is in food and hygiene. Bacteriophage are used to control and eliminate bacterial contaminations or their population from the food, and are also highly specific, which makes them attractive for sanitation and of ready to eat food like milk, vegetables, etc. Fourth is in agriculture. Some bacteriophages that are specific to plant bacteria have found their application in agriculture. The phages are used for the treatment and prevention of bacterial diseases in plants. The use of bacteriophage in place of antibiotics prevent the use of clumping of antibiotics on the plant surface, which might be harmful to the health of consumers. These are the references which I have used, and thank you. No, no, sir. No, sir, it was that only it was written. Other than, okay, the other than lambda phage. Uh, it is T2, T4 phages. Phage lysate. Yes, sir, yes. I don't know. So, the genetic materials inside the head, which is made up of हाँ.
Yes, sir. Yes, sir. Is my presentation visible? Hello? Okay, so today we are going to discuss about artificial chromosome, and in artificial chromosome mainly about bacterial artificial chromosome. So artificial chromosomes are vector which can contain a large segment of DNA, and that DNA, we can study that large segment of DNA. We can study that a large segment of chromosome with help of different techniques that are available. That are techniques like, okay, whatever. Sorry, sir. Sir, can I get 5 seconds? Okay. So artificial chromosomes are like we create a plasmid-like structure in which we can insert a large segment of chromosome, which we can use to study, like in research purposes. And artificial chromosome can contain an entire genome in a manageable number of clones. Like we can create as we can create many copies of the required chromosome. Uh, so bacterial artificial chromosome is engineered DNA molecule used to clone DNA sequence in bacterial cells. So bacterial artificial chromosome, we create that bacterial artificial chromosome, and the cloning of that DNA molecule is done inside bacterial cell, and for this, we can use the bacterial cell of E. coli, bacteria like E. coli. Uh, so the segment for of an organism's DNA ranging from 10 kilo base pair to about 30 kilo base pair can be inserted into bacterial artificial chromosome. Uh, Segment of organism's DNA size of insert. Segment of an organism ranging from 10 kilo base pair. नहीं, 100 kilo base pair. Sorry, 100 kilo base pair to about 300 kilo base pair can be inserted into bacterial artificial chromosome. Uh, the bacterial artificial chromosome with their inserted DNA are then taken up by bacterial cells by techniques which we will see further. As the bacterial cells grow and divide, they amplify the bacterial artificial chromosome DNA, which can be then isolated and used in sequencing of DNA. So, I would like to explain it with help of this diagram. So here
we for making a bacterial artificial chromosome, we take the gene of interest. We take our gene of interest, and we attach the ori site. No, we insert the gene of interest into a bacterial plasmid, and then through techniques such as electroporation, etc., we induce the bacterial artificial chromosome into uh, bacteria such as E. coli, and after inserting the artificial chromosome in bacteria, this bacterial artificial chromosome, as it has ori site, it has multiple cloning sites. So it replicates in this bacteria, and we get colonies having those characteristics, showing the characteristics of that chromosome. And after we insert this artificial chromosome in a bacteria, we can detect its presence with the help of screening method like blue-green screening. So here we take an example of this bacterial artificial chromosome, P. bello, blue-white, sorry, blue-white, blue-green name. P. bello, bacterial artificial chromosome 11. So, uh, there are a set of criteria which must be followed for making this artificial chromosome. First is it must have a selectable marker, like in this artificial chromosome. Here we have a selectable marker of this CAM-R gene, that is chloramphenicol resistant gene. So, what the use of this resistant marker is, so when we will insert this artificial chromosome in a bacteria, and when we will plate it, so like suppose we have this artificial chromosome, when we will plate this newly induced bacteria into a media uh, having chloramphenicol, the uh, बैक्टीरिया जिसमें सक्सेसफुली आर्टिफिशियल क्रोमोसोम इंड्यूस हुआ है वो ग्रो करेगा. सो वी विल नो दैट आर chromosome is inserted into the bacteria. And multiple cloning site is helpful so that हमें ज्यादा number of copies मिले. इसमें मल्टीपल क्लोनिंग साइट लैग जेड प्लस के जगह पे आना चाहिए था, बट एज ओरिजिन साइट हम लोगों के पास ऑलरेडी है, और ये वाला जो आर्टिफिशियल क्रोमोसोम है वो एफ फैक्टर जो ओरिजिन साइट है उसके हेल्प से मल्टीप्लाई करता है. सो यहाँ पे वी डोंट हैव मल्टीपल क्लोनिंग साइट, and this particular vector can be used with the blue-white colony screening method. A large piece of DNA can be engineered in a fashion that allows it be propagated as a circular artificial chromosome in bacteria, so-called bacterial artificial chromosome. तो हम लोग उसे ऐसे डिजाइन करते हैं कि लाइक सर्कुलर फैशन में आ जाए हमारा आर्टिफिशियल क्रोमोसोम. Each bacterial artificial chromosome is a DNA clone containing same point 100 to 300,000 base pair, that is kilo base pair. Bacterial artificial chromosome is much smaller than endogenous bacterial chromosome. It is straightforward to purify the bacterial artificial chromosome DNA away from rest of the bacterial cells DNA, and thus have cloned DNA in a purified form. So जब हम लोग ये आर्टिफिशियल क्रोमोसोम बैक्टीरिया के अंदर इंड्यूस करते हैं, तो अगर वी वांट दिस आर्टिफिशियल क्रोमोसोम सेपरेट, तो लाइक हम लोग सेल का लाइसिस करवाते हैं और उसके अंदर से जब हम लोग डीएनए एक्सट्रैक्ट करते हैं, तो उस प्रोसेस में भी हम लोग बैक्टीरियल आर्टिफिशियल क्रोमोसोम को एक्सट्रैक्ट करना इज वेरी मच इजी इन बैक्टीरियल सेल. Bacterial artificial chromosomes do not undergo rearrangement in host. Therefore, they have become the preferred vector for making large colonies in physical mapping studies of genome. तो मतलब वो सीक्वेंस वो बैक्टीरिया के अंदर जाके मॉडिफाई नहीं होता. Bacterial artificial chromosomes are often used to sequence the genome of organism in genome projects. For example, human genome project. सो जब ह्यूमन जीनोम प्रोजेक्ट हुआ था तब लाइक हम लोग ह्यूमंस के लाइक बहुत सारे कॉपीज बनाए थे, राइट. तो वो एक एक सेगमेंट हम लोग आर्टिफिशियल क्रोमोसोम इंड्यूस करा रहे थे. वो बैक्टीरियल आर्टिफिशियल क्रोमोसोम क्रिएट करके उसका लाइक सीक्वेंसिंग हम लोगों ने इजी कर दिया था. So disadvantages of bacterial artificial chromosomes are as follows. AT-rich DNA fragments. DNA fragments with high proportion of A and T nucleotides typically do not clone well. DNA sequences are toxic to E. coli and hence are unclonable in that organism. So जब हम लोग ये बैक्टीरियल आर्टिफिशियल क्रोमोसोम ई कोलाई में insert करते हैं तो it is toxic for for E. coli. Applications: study of large double-stranded DNA virus for academic research and advanced vaccine genomic libraries, basic science research to industrial research like animal husbandry, phylogenetic lineage between different species, study of horizontal gene transfer. These are the references. Thank you, sir. नो सर, इट इज, बट इस केस में मल्टीपल क्लोनिंग सेट यूज नहीं होता. एक्सेप्शंस: इंसर्शन, इनएक्टिवेशन. नो आइडिया सर. सर जो आपने बोला इंसर्शन इनएक्टिवेशन, डज इट हैज टू लाइक रिलेट समथिंग लाइक हम लोग वो चीज इंसर्ट करते हैं और फिर जो उसका फंक्शन है वो परफॉर्म नहीं होता है इसलिए वो कुछ इनएक्टिवेशन सॉर्ट ऑफ हो जाता है. नो. थैंक यू सर.
सर वो नहीं देगी, उसको फीवर है, वो कल दे देगी.
Yes, sir. Yes, sir. Yes, sir. Sir, is it visible? Good evening. My name is Rizwan. My topic is shutter vector. So, first we will see what is a vector. Vector is a DNA molecule, often plasmid or a virus, that is used as a vehicle to carry a particular DNA fragment into host cell as a part of cloning or a recombinant DNA technique. There are many types of vector, such as expression vector, shutter vector, PCR cloning vector, transcribable vector, non-plasmid vector, and many more. But as my topic is shutter vector, we will get into it. Shuttle vector is a vector that can be introduced into two or more different host organisms and maintained by either of those organisms. So what is the general properties of shuttle vector?
These vectors can be designed to replicate in cells of two different species. Therefore, they contain two origins of replication, one specific for each host species, as well as genes necessary for the replication. These vectors are created by recombination technique. Some of them can be grown into different prokaryotic species, usually E. coli, and a eukaryotic one example is plant cells or animal cells. YEP13 is an example of a shuttle vector. So YEP13 is a YEP type Saccharomyces cerevisiae coli shuttle vector for cloning specific genes by transformation of yeast. Its YEP contains 2 micrometer origin of replication. The 2 micrometer plasmid is an excellent basis for cloning vector. Replication makes use of the plasmid origin. Several enzymes provided by the host cell and protein encoded by rep1 and rep2 genes carried by the plasmid. In this, Luke2 acts as a selectable marker, which codes for B-isopropyl malate dehydrogenase, one of the enzymes involved in the conversion of pyruvic acid to lysine. In order to use LIKE2 as a selectable marker, special kind of host organism is needed. The host must be an exotrophic mutant that has a non-functional LIKE2 gene. It also includes entire PBR322 sequence and can therefore replicate in the selected and can be selected for both yeast and E. coli. Thank you. Uh, no, sir, I'm not showing the diagram. No, sir. It can be used for the transferment for yeast, where you can grow in two different organisms at the same time and see the difference. Like you don't have no like in it can transform like from one species, yeah, from eukaryote and prokaryote. You can transfer it like for other vectors. You cannot like use in prokaryotic plasmid or vectors in eukaryotic. For this, you can use on both of them. So you can engineer it in E. coli and like transfer or transform the host cell as like yeast and that. Two. One for E. coli, one for yeast. I don't know, sir. Mm, no, no, sir. Yes. Mm, I don't know, sir. Thank you, sir.
Yes, sir. Yes, sir.
So good evening. Sir, is my screen visible? Okay, so good evening everyone and all. My name is Smita Garud and my roll number is 227309. So today I am presenting a topic, expression vector. So we all know that what is vectors. Vectors are a DNA base which carry the sequence of DNA or other genetic material. So what are expression vectors? Expression vectors are usually plasmid or virus that are specially designed for expression of genes. So the vector is usually used to introduce a specific gene into a target cell. Expression vector are basic tool in biotechnology for production of protein. So for expression vector, there are some features that must be available in vector. So one is promoter of multiple cloning sites, transcription terminator at downstream, DNA sequencing coding like Shine diagram sequence for initiation of translation. So here is the ideal vector, ideal expression vector which has a promoter at upstream, terminator at downstream, multiple cloning site, Shine diagram sequence for initiation of translation, ORI, and the selectable marker that is ampicillin resistant. So what is basically it is a cDNA of our gene of interest. So this cDNA is inserted at a cDNA is inserted at a recognition site H1. So once it is inserted, it the after the once it is inserted, once it is inserted in our cell, that cell is undergone transformation and the transcription is initiated. And after the transcription, with the help of Shine diagram sequence, translation is begin and translation produces polypeptides encoded by our gene of interest. So expression in E. coli, there are some advantages and drawbacks also. The advantages are rapid doubling type, growth in simple and defined media, extensive knowledge of its promoter and terminator sequence, protein of both prokaryotic and eukaryotic origin can be produced within the organisms. Many proteins of prokaryotic are originated in E. coli due to E. coli cells are easily broken for the harvesting of protein produced within the cell. Drawbacks are E. coli cells are unable to process introns or do not possess the extensive post-translation machinery like glycosylation, methylation, phosphorylation, etc. So there are two types of expression vector, a prokaryotic expression vector, which is bacterial expression system, which has LAC promoter, TAC promoter, APL promoter, and T7 expression system. Eukaryotic expression vector have yeast expression system, that is Saccharomyces cerevisiae, viral expression system, that is baculovirus, and mammalian cell expression system. So now we are going to see the properties of LAC-Z promoter. So LAC-Z promoter operator is in a frame with LAC-Z alpha fragment, the NH3 terminal part of galactosidase gene. Multiple cloning sites are found in border of NH3 including ATG sequence. The presence of such restriction site sequence should not disturb the functional activity of the protein, which complement with the omega fragment of LAC-Z produced by the bacterial cell as a complement. If any gel is placed in a proper frame in the NCS, the protein expressed will be fused form. The expression of the gene can be regulated by isoprobate thio-based galactosidase, that is IPTG. So the applications of expression vectors are the construction of expression libraries, the analysis of gene function at protein level, the commercial production of proteins, the production of antibodies, or for in vivo study of proteins. And here are the references. Thank you, sir. Yes, sir. कहाँ कहाँ पर सर, ये वाला डायग्राम है, ये वाला डायग्राम, ये वाला? हाँ. हाँ सर, वो रिवर्स डायरेक्शन में भी इंसर्ट हो सकता है, लेकिन जब ट्रांसलेशन स्टार्ट होगा तो हमें पता चलेगा कि प्रोमोटर से स्टार्ट हो रहा है कि टर्मिनेटर से स्टार्ट हो रहा है, क्योंकि जो वहाँ H1 साइड पे वो जो इंसर्ट होता है, तो वो साइड दो पार्ट में डिवाइड होता है, वन प्रोमोटर और टर्मिनेटर। तो ट्रांसक्रिप्शन स्टार्ट कहाँ से होगा, उससे पता चल जाएगा सर। नहीं, एक्सप्रेशन वेक्टर में होता है सर, प्रोमोटर एंड टर्मिनेटर। गलत आंसर। सर मैंने जितना पढ़ा था मैंने उतना बोल दिया सर। हाँ, ये वाला। सर शाइन डायग्राम सीक्वेंस से जो ट्रांसलेशन इनिशिएशन के लिए रिक्वायर है, और भी बताना है सर। मतलब क्या बताना है सर? सर वो ट्रांसलेशन, सर ट्रांसलेशन इनिशिएशन। इतना इतना ही याद आ रहा है सर अभी। ट्रांसलेशन इनिशिएशन के लिए यूज होता है। वीडियो से स्टार्ट हो रहा है सर। सर इन विवो स्टडी करने के लिए प्रोटीन का, फिर एंटीबॉडी प्रोडक्शन के लिए, फिर एक्सप्रेशन लाइब्रेरीज के लिए। थैंक यू सर।
Yes, sir. Okay, sir. So, good evening, everyone. My name is Vedanti. My roll number is 227310, and the topic which I am presenting is cosmid. Sir, next. So, what are cosmid? Cosmid vectors are hybrid between the plasmid and fast lambda vectors. So, cosmid have cos sequence and lambda patch. So, the first cosmid vector was described by Collins in 1978. These vectors are developed by combining the features of plasmid vector and the bacteriophage vectors. So, some example of cosmid vectors are, the classic example of cosmid vector is C2RB, and another example is PHV79. It's one of the most common cosmid used in genetic engineering. Next. So the structure of cosmid, so it has origin of replication, which is basically ori, and this is a restriction site for the cleavage and the insertion of foreign DNA. uh, it contains selectable marker from the plasmid also. It could be antibiotic resistant marker, and it has a cos site. A sequence is cohesive end. So this cos site is 200 base pair long, which helps to uh, which helps in linearization of a DNA. Cosmid. So, what are the important features of cosmid? Cosmid are plasmid that contain a cos or a cohesive end uh, that are a sequence required for the packaging of DNA into lambda phage particles. Cosmid also contain a phasmid origin of a DNA replication, at least one selectable marker, and useful restriction endonuclease sites. Next, here is a diagram where we can see. So next, so the properties of cosmid. Cosmid are medium-sized cloning vectors. Their transformation frequency is high. Their cloning capacity is 35 to 45 kilobase pair, where normal plasmid can only carry 1 to 20 kilobase pair. They replicate as a plasmid if they have a suitable origin of replication. They frequently also contain a gene of selection such as antibiotic resistant. A cosmid vector may have one or two cause sites. The next, then cloning of foreign DNA in a cosmid vector involves the following steps. So first, ligation of foreign DNA between two cause sites, making a con- contameric DNA, then in vitro packaging to introduce a DNA into phage head to form the mature phage particle, then introduction of the clone DNA into E. coli by transduction. After their entry into a host cell, the cosmid are maintained as plasmid. The next, then here are some advantages of cosmid. They can be used as a gene cloning vector. They They have they are high insert capacity vectors. They are used for the analysis of complex genomes. Large gene can be studied intact, and genetic linkage studies can be carried out at the molecular level. Then transformation frequency of cosmid is high. They are useful for cloning very large DNA fragments, 35 to 47 kilobase pair, in the site selection for the large insert. We can handle them like plasmids. Next. So there are drawbacks also. This approach is very sensitive. Like we have the exact ratio of DNA insert to vector DNA because vector to vector ligation can occur. And recombination plasmid have duplicate vector sequence are unstable during cloning. Next, some application of cosmid. We get in cloning, gene manipulation, pharmaceutical and medical industries, genetic recombination, and in genomic libraries. Next, thank you. Okay, CLO cloning. एक मिनिट. CLONNING. सिंगल एन. नहीं. सिंगल एन. गलती से हो गया सर. हाँ. Because it its carrying capacity is 35 to uh, 45 kilobase pair. Plus, it has cross site which has sticky ends, means cohesive ends. So, flanking overhang is there. Carrying capacity ना सर जो नॉर्मल तो हाँ select efficiency क्योंकि इसमें कॉस्ट टू कॉस साइड रह सकते हैं, रेजिस्टेंट, क्योंकि ये इसमें पैकेजिंग में मतलब फाज में पैकेजिंग में फाज के हेड में पैकेजिंग में इजीली हो जाता है, मतलब फिर मल्टीप्लिकेशन के लिए यूज होगा ना सर, मतलब हाँ क्या डीएनए मतलब कंटिन्यूअस डीएनए मतलब लॉन्ग मॉलिक्यूल जिसमें मल्टीपल कॉपीज होंगे डीएनए सीक्वेंस के। हाँ ओके थैंक यू सर।
Yes, sir, I am there, but can I present tomorrow, if possible? Thank you, sir.
Yes, sir. Yes, sir, I'm ready. Sir, is my screen visible? Okay, okay. So good evening, everyone. My name is Sakshi Guru. My roll number is 227312, and the topics of my presentation are PCR cloning vectors and transcribable vectors. So let's see the first topic, that is PCR cloning vector. So as we all know what is PCR, so PCR is basically used for the amplification or cloning of the DNA. So what sorry sir, sorry for the disturbance. Sir, okay. So, what happened is it is possible to clone the DNA segment that are blunt end. But what happens in PCR is, in PCR, as we use a thermostable enzyme, that is Taq polymerase. So what Taq polymerase does, it do not creates an blunt ended DNA. So it creates an overhang at 3' end, and that overhang is formed such that there is an means that enzyme adds an unpaired A nucleotide, that is adenine, at the 3' end. And to overcome this, so as there is an unpaired means blunt ends are not formed, so it is impossible to clone the that DNA. So to overcome this, the vectors are designed such that the vectors are delivered with T nucleotide that has a overhang at 5' end. So to that this process is known as TA cloning, that is T stands for thymine residue and A for adenine. And how this process works. So first step in this is the creating the insert. So as I told, so during PCR the Taq polymerase will create an overhang at 3' end, and after that for its cloning we will be creating the vector. So how this vector is prepared, so vector is first linearized with restriction enzymes, and it is stayed with DDTTP, that is dideoxythymidine triphosphate. And this DDTP enzyme will add T nucleotides at the 5 prime end, and it will create a 5 prime T overhang residue by a mechanism known as terminal transferase. Means it will be adding the T group, T nucleotide at the terminal end, and it will create a non-sticky end. Now, as we have the insert that has an A nucleotide overhang, and we also has a plasmid that has a T nucleotide overhang. So now there is that we have to join those insert and plasmid. So we have to ligate, and for this ligation, DNA ligase enzyme is used. So this DNA ligase enzyme will join the vector and the insert by forming the phosphodiester bond between 3 prime OH and 3 prime OH of the adenine residue and 5 prime phosphate group of the plasmid. So in the diagram you can see that in the insert we have an A overhang, and after that we will insert this insert into a plasmid that has a T overhang at 5 prime end, and after that a plasmid, a full form plasmid will be formed after insertion by using DNA ligase. And further, the as we have now the whole sequence of the DNA, so we can be using that for the further cloning. So my next topic is transcribable vector. So a transcribable vector is a plasmid vector only, which has a promoter for an RNA polymerase. And these transcribable vectors are designed for the transcription of insert in vitro as well as in vivo. And the promoters which we are using is basically from bacteriophages such as T7, T3 or sp6. So in the diagram, you can see plasmid is there, and in plasmid we have inserted a gene of interest. The gene of interest means the one which we have to transcribe. So the promoter here is T7 promoter, which is present at just near the multiple cloning site. And after that, the gene of interest is then inserted into the plasmid. Then as there is a T7 promoter, so we will be adding T7 RNA polymerase only, that is specific RNA polymerase will be adding. Then we will be adding NTPs, and then we will be transcribe. Then there will be transcription of this gene of interest, and you can see the full form mRNA is then transcribed. This is in vitro process, and beside there is an in vivo transcription. So for here it is given in the diagram that in E. coli it has a gene of interest that has to be expressed, and that gene of interest has a T7 promoter only. But for T7 promoter, we do not have T7 RNA polymerase. So for that, what we are doing, we are taking a plasmid vector. In that, we are inserting a gene that expresses T7 RNA polymerase, and then this plasmid that contains T7 RNA polymerase gene is then inserted into the E. coli by a transformation process. And how this T7 RNA polymerase is expressed, the it will the host RNA polymerase will transcribe this T7 RNA polymerase gene, and on transcription and translation, this T7 RNA polymerase is expressed in the E. coli, that is in the host. And now this T7 RNA polymerase, which is expressed, will be uh, then will be functioning with the T7 promoter of the host cell, and it will translate those that gene of interest which we have to express in the E. coli. So this if we are doing this reaction in vitro, we will be adding T7 RNA polymerase, then NTPs and buffer, and this reaction is incubated at 37 degrees Celsius. So this is how the mRNA which is not transcribed fully will be transcribed by using the these vectors. So that's it. I have referred iGenetics for it. Thank you, sir. Yes, sir. No, sir, because we are using plasmid. So yes, sir. हाँ सर, cDNA का ही रिक्वायरमेंट है क्योंकि पहले वो ट्रांसलेट होने के बाद ही हम यूज कर सकते हैं. सर क्योंकि कॉम्प्लीमेंट्री चाहिए ना, सो फिर वी विल बी यूजिंग कॉम्प्लीमेंट्री ओनली जीनोमिक. सर आप फिर से सर आप प्लीज फिर से क्वेश्चन एक्सप्लेन कर दो. हाँ सर. हम्म. हाँ सर, वो सेल्फ नॉन सेल्फ. सॉरी सर, अब याद नहीं आ रहा है. हाँ. हाँ सर, सिस्टम अलग है इसलिए. हाँ सर, न्यूक्लियोटाइड्स. सॉरी सर. Sir, it is deox. um, sir, mm, वो क्या बोलते हैं उसको, क्योंकि that will add nucleotides. That will be sir means वे सर आ रहा है पर एक्सप्रेस कैसे करूँ वो नहीं समझ. सर मतलब हम elongate करते हैं ना, वो चेन elongate. हाँ सर, इतने. हाँ सर, इतना ही है ये। थैंक यू सर। हाँ 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 सर, स्पेसिफिक ही यूज़ करेंगे। एंड मीडिया का नाम नहीं पता, पर but means it will express now, so we'll be using such media, selective media we'll use. The हाँ marker, हाँ sorry sir।
सर कल प्रेजेंट करती हूँ।
Yes, sir. Yes, sir. Yes, sir. So is my screen is visible? Sir, up? Hello everyone, my name is Mansi Director and my topic of presentation is chromosomal library. So what are chromosomal libraries? A library consists of a collection of a cloned DNA fragments derived from one chromosome is called a chromosome library. So a chromosome library, 24 different libraries for the 22 autosomes, the X and the Y. Since each chromosome is far smaller than the total genome, the resulting libraries can also be smaller. Using these chromosomal libraries can simplify, we can simplify later organization steps as the genomic sequence is assembled because all the clones in each chromosome library are by definition from the same chromosome and thus from the same large pieces of DNA. मतलब हम लोग यहाँ पे क्या बोल रहे हैं कि जो मतलब ऐसे ह्यूमंस में 46 क्रोमोसोम्स रहते हैं और 23 पेयर्स में रहते हैं, तो 22 ऑटोसोम्स रहते हैं और जो 23rd वाला रहता है वो 6 क्रोमोसोम रहता है, जैसे एक्स एंड वाई। तो जो तो ये लाइब्रेरी जो क्रोमोसोम लाइब्रेरीज होते हैं उसमें क्या होता है, वो वो डिफरेंशिएट किए जाता है। जैसे कि जो फर्स्ट क्रोमोसोम होगा वो सबसे लार्जेस्ट क्रोमोसोम होगा और जो 20 फर्स्ट वाला होगा वो स्मॉलेस्ट क्रोमोसोम होगा। यही यहाँ पे कहा गया है। और अभी इम्पोर्टेंस ऑफ क्रोमोसोमल लाइब्रेरी, these libraries proved to be quite useful in certain aspects of the human genome project. As several research teams had been assigned specific chromosomes to sequence, and they turned to these smaller, less complex libraries to make their analysis simpler. If you use to clone a specific gene but do not have genomic sequences, libraries, either genomic or chromosome, will be important tools for finding and cloning that gene. तो ये जो लाइब्रेरीज है क्रोमोसोमल लाइब्रेरीज वो ह्यूमन जीनोम प्रोजेक्ट में बहुत यूजफुल थी. ह्यूमन जीनोम प्रोजेक्ट बेसिकली ये था कि उसमें वो सबसे बड़ा प्रोजेक्ट था और उसमें साइंटिस्ट ने प्लान किया था कि जो ह्यूमन का जीनोम है उसको वो ह्यूमन जीनोम का सब सीक्वेंस स्टडी स्टडी करने का प्लान था. तो ये जो लाइब्रेरीज है वो उन जैसे प्रोजेक्ट में बहुत हेल्पफुल रहती है. So how our libraries are made. Individual chromosomes can be separated if their morphologies and sizes are distinct enough, as is the case for human chromosome. In one separation procedure, flow cytometry, chromosomes from cells in mitosis are stained with a fluorescent dye and passed through a laser beam connected to a light detector. This system sorts the chromosome based on the differences in the dye intensity that result from certain differences in the abilities of the various chromosomes to bind the dye. Once the chromosomes have been sorted and collected from several cells, तो बेसिकली यहाँ पे ये मतलब बोला गया है कि जो जो क्रोमोसोम जैसे जैसे मैंने पहले बोला कि जो फर्स्ट क्रोमोसोम होगा वो सबसे लार्जेस्ट होगा और जो 21st जो क्रोमोसोम होगा वो सबसे स्मॉलेस्ट होगा. तो वो उसके तो किस बेसिस पे हम बोल रहे हैं कि जो लार्जेस्ट होगा तो वो मे भी उसका डीएनए का जो साइज हो सकता है वो सबसे बड़ा हो सकता है, इसलिए वो सबसे लार्जेस्ट है जो फर्स्ट क्रोमोसोम है. तो उसको हम लोग अगर डिफरेंशिएट हम लोग कैसे करते हैं तो वो एक प्रोसीजर है फ्लो साइटोमेट्री करके. उसमें हम लोग उसके बेसिस पे वो प्रोसीजर यूज करके या वो टेक्निक यूज करके हम लोग ये क्रोमोसोम को उनके साइज साइज के हिसाब से हम लोग डिफरेंशिएट कर सकते हैं. सॉरी. हाँ. तो इसमें जो जो लार्जेस्ट क्रोमोसोम होगा तो फ्लोरेसेंट जो हम लोग डाई जो उसमें ऐड करेंगे जो लार्जेस्ट होगा उसके जो तो उसकी इंटेंसिटी ज्यादा होगी. तो इसके हिसाब से हम लोग लाइक वो हम लोग जान सकते हैं कि कौन सा लार्जेस्ट है और कौन सा स्मॉलेस्ट. टाइप्स ऑफ क्लीवेज. तो ये क्रोमोसोम लाइब्रेरी का एक्चुअली में जो मेथड है मतलब वो कैसे बनते हैं. तो यहाँ पे बेसिकली क्या हुआ है साइट ऑफ क्लीवेज और ये डीएनए है. उसमें 4 फ्रैगमेंट्स है, फ्रैगमेंट 1, फ्रैगमेंट 2, फ्रैगमेंट 3 और फ्रैगमेंट 4 ह्यूमन डीएनए है. उसमें उसको रिस्ट्रिक्शन एंजाइम के हेल्प से कट किया है और फिर रिकॉम्बिनेंट डीएनए फिर हमने बनाया है. तो जो हमारा जो फ्रैगमेंट है उसको प्लाज्मिड में इंसर्ट करके जीन फॉर रेजिस्टेंट टू एंटीबायोटिक एंटीबायोटिक का रेजिस्टेंस वो जीन है. तो उसमें हम लोगों ने वो डीएनए डाला है और फिर ट्रांसफॉर्मेशन हुआ. तो बैक्टीरिया में इंसर्ट करने के बाद ट्रांसफॉर्मेशन के बाद जो बैक्टीरिया विदाउट प्लाज्मिड है दे फेल टू ग्रो प्लेट विद एंटीबायोटिक कंटेनिंग मीडियम और जो बैक्टीरिया विद प्लाज्मिड लीव एंड मल्टीप्लाई प्रोड्यूसिंग जीनोम और क्रोमोसोमल लाइब्रेरी. Only a small part of the one chromosome is shown. A great many more DNA fragments would have produced from an entire chromosome or genome. Applications. Chromosome analysis is a test to look at the chromosome in a sample of cells. It can help identify genetic abnormalities as the cause of a condition or disease. The test can count the number of chromosomes present and look for any structural abnormalities in the chromosome. तो ये क्रोमोसोम डीएनए लाइब्रेरी में क्रोमोसोम डीएनए लाइब्रेरी क्या है एग्जैक्टली वो है एक्सप्रेशन. ओनली स्पेसिफिक जीन्स होते हैं उसमें. पूरा जीनोम नहीं होता जैसे जीनोमिक लाइब्रेरी में होता है वैसे पूरा जीनोम यहाँ पे नहीं होता. ओनली स्पेसिफिक जीन पे होते हैं. साइज स्मॉलर रहता है कंपेयर टू जीनोमिक लाइब्रेरी. इंट्रॉन्स एब्सेंट होते हैं. एक्सॉन्स होता है सिर्फ और वेक्टर्स इट हैज नो इंट्रॉन्स. So uses plasmids, fragments, lambda phases to accommodate small fragments.
Thank you, sir. ओके, तो मुझे कल फिर से मतलब प्रेजेंट करना पड़ेगा क्या सर? ओके ओके फाइन. ओके. थैंक यू सर.
Thank you, sir. Bye.
Bye, sir. Good night.